Thank you for introduction. I'm Myungwan Choi from Seoul National University. As introduced, we are gonna speak by three presenters. And the title of this talk is Towards Biological Communication with Light. So as you can see in this video, neurons communicate with electricity. So neuron generates action potential in their cell body, and the action potential travels through long projecting axons. And this transmission speed can go up to about 100 meters per second, which forms the fastest communication mode in biology. So here we thought about how can we overcome this biological limitation. So obviously the fastest form of communication is light, which can go at 3 times 10 to the 8 meter per second, which is a million times faster than the biological communication. So we wanted to develop optically communicating neural network. So to implement optically communicating neural network, we think the basis would be generating optical synapse. So this optical synapse is composed of two parts. One is light emitting neuron on the left, which is the presynaptic neurons, and light receiving neurons on the right is the postsynaptic neurons. So light emitter neurons generates their spontaneous activity and electricity, voltage, and we wanted to convert that into light. So to read this neural activity, we use calcium indicator because neural voltage activity is biologically converted to fluctuating calcium ion signal in their soma. So this calcium light, but it was not really enough to induce postsynaptic activation. So we wanted to amplify this light level, and also we wanted to mimic action potential like output, which is highly nonlinear, all or non-like behavior. So to implement that, we use this activity dependent lasing. So we are employing micro laser into here to amplify and also thresholding the light output. This output is relayed by this optical fiber, which can transmit light losslessly over very long distance. And the light receiving part is, as well introduced by Francesco, is optogenetics, by optogenetics. So we transduce these neurons to express optogenic actuator tuned to this lasing light. So basically, by combining this existing technology, we aim to develop this optical synapse. So this is our team. So Matt has expertise in micro lasers. He can turn anything into a laser. So he was in charge of converting neurons into emitting laser light. And I was in charge of making genetically engineered neurons and neural circuits. And Hyung-soon is in charge of generating a whole system into a on-chip device. So a little more detail. For light emitting part, we culture neurons, we culture primary neurons, and transfecting those with calcium sensors, and we put them into the laser cavity. This laser cavity is formed by these two dielectric mirrors, and there's a pump laser to amplify the light, and this light output, laser light, is coupled to optical fiber or transmit by free space to activate the postsynaptic light receiving neurons. For light receiving neurons, we prepare neurons transfected with this optogenic actuator, C1V1, which can be activated by green light. And here we are testing if they can be activated by the laser light. So here we are, for recording, we are using voltage indicator, burst 1. So we're recording voltage here, and we are giving well-defined light input using DMD, which we use it for a projector. And we found that these neurons can be activated by down to 1.1 milliwatt per millimeter square, which Matt can generate with a cell laser. So I'll give my microphone to Matt, and he will continue discussing about the micro laser.
Yeah, so like Mark said, so we need this 0.1 milliwatt per square millimeters, which sounds small, but it's quite large intensity. So we need to generate this within a biological laser. So what I mean by that, in my lab we work with very special lasers. So this work started almost 10 years ago when we made the first laser inside a live cell. we just put these fluorescent bits to the cells, they uptake them, and we have this bit inside. So it's a laser inside the cell. And we can use these biological lasers for a number of different applications, including barcoding. So we can barcode the cells and track them. We can do also this like in food to make edible lasers and put barcodes in like wine or some food. Then we can do a number of different sensing, because these lasers are very sensitive to the environment. We can do, for example, sensing of the size change of lipid droplets or like forces within tissues. And then we can also implant like in tissues, blood, and so on, and do various kinds of biological imaging by using these micro lasers. So how a laser works, yeah? So here I compare just normal fluorescence and a laser. So normal fluorescence, you all know, we excite with a short wavelength, and then this emits in a longer wavelength with a broad spectrum, and also the output of just fluorescence is linear dependent on the input. But how we met a laser is just that we put this any kind of fluorescent material inside the laser cavity. So, for example, 2 mirrors or a droplet or a sphere, and now the light is trapped inside and is amplified. And in this case, we then have a threshold behavior. So below the lasing threshold, we also don't have any light, but afterwards we have high emission. So this is kind of similar effect as in threshold behavior in a neuron, right? And also the emission spectrum is very narrow, and we have the directionality. So we can direct this light from the cells to the other cells. And specifically how a cell laser is made, you have 2 laser mirrors, dichroic mirror, and inside there are cells which either express fluorescent protein, or we provide some fluorescent dye. And we excite with external light, and there is emission of laser light. And this is an image of a cell that lases and spectrum, and we have this lasing threshold. So how to combine this for the optical communication is that we have some kind of a sandwich structure, so not with optical fibers yet. Yeah? So this is just the beginning, and you have the 2 or 3 mirrors here, and the upper part there are like cells, or initially we just use dye, fluorescent dye to test this, and at the bottom there are these cells that receive this signal, and then with another laser we then do calcium imaging to see if we can really activate these cells. This has to be designed quite carefully because we have a number of wavelengths that we are using for excitation. Then this is in green for excitation of cells, and then we need to do calcium imaging. At the end, Hyun will talk about how this will be integrated together. And just as some first results is that with different dyes, actually not with cells, we also work with cells, but with dyes we already get high enough power so that like in 2 weeks in Korea we will try to actually do the cell-to-cell optical communication for the first time. And now I give word to Hyun-sun.
All right, just give a little bit background. So I'm an electrical engineer working in a hospital. So when I heard this idea, I found this really, really interesting. But 10 years forward, thinking about it, how we can make really circuit, or we can make optical communicating like a circuitry. The idea is it's not just for the single cell to another cell. We need to make an arrays of the cell or their network to make it really generate this optically communicatable circuitry. So my idea was we want to make this single cell capture site. So we want to arrange this cell and the colony in a specific area. And then we want to make a portable imaging system to continuously look at these cell behaviors. More importantly, we want to make this programmable for fully automatic fluidic control, such that the cell can be stimulated with the nutrition and the different chemical reagent, and then we can continuously monitor their activities. So we let the cell do their work, and then we take a piece. So for the imaging system, we have been developing this portable imaging cytometry system. So when you think about the imaging cytometry system, it's a very bulky, expensive system. Or you also think about like a multi-channel fluorescence imaging system with a fully automatic operation that is also very bulky and then difficult to integrate with the other circuitry. So the idea is that we want to make a very small, like about 30 centimeter cube imaging system. So initially we start with a 4 imaging channel. A couple of years ago, we developed the upgraded version having a 6 imaging channel. So in this small box, everything is fully automated, and then you can take multiple images. And actually, this is the one example of showing the tissue section, staying with the 12 different colors, and then we can easily get a really good quality of these images from this small box. So when we compare this quality of the images with the conventional microscope, we see the very equivalent level of the performance. So the idea is we want to use this portable imaging system to facilitate this integration of the optical communication circuit. The last part is the latest version is that we want to combine them in an on-chip integration. As Matt explained, and we want to have a multi-stack layer, and then we want to capture this cell in a specific area, and then we activate them and then look at their optical signal changes. And during the discourse, we come up with this microfluidic component and the control system such that we can automatically program what we want to do. We want to inject a specific solution, and then we can stop, we can adjust the speed and the time. So it's very flexible. Like we can introduce up to 12 different solutions, and the entire procedure can be fully automated with our program. Then we can choose what solution we want to flow rate, and the duration. And then through this microfluidic channel, we can actually expand this concept, not only for the single cell to single cell, we can actually make arrays and then having their interaction and then monitor them in the multicolor imaging. So, I mean, you can imagine that through this conference, we discuss a lot of different ideas. I mean, you may find other application using this kind of system. So actually, we are applying this system for other, like a cellular analysis in the different various bodily fluids, such as CSF, lung fluid, and ascites, depending on what you are interested in. So this is the image of the like a cell captured in our microfluidic channel. You can see the number of cells we can capture. And then I can zoom in, we can actually get those multiple color images from each individual cell. So as Matt mentioned, in 2 weeks in Korea, 3 teams will gather together, bring each component, the laser and the fluidic component, and then we'll do the first integrated experiment. So we are very excited about this opportunity, and then I think this is just starting of our journey. And then, but as you can see, this is a very unique team of the physics, neuroscientists, and engineer to realize this unheard of, the very interesting idea. So I really thanks for the Human Frontier Science Program to support this research. And hopefully after the 2 weeks, we can get the more interesting data and then share the result in the next time. And then here at the HPI's lab, and then we are open to any other discussion as well. And then, I mean, I'm like the only one having a big scar in the forehead, so you can easily find me right here. And then, yeah, thanks for your attention, and happy to take any question. Thank you.
Hello. Thanks for the great talk. I was having a question about the optical setup, so part 2. And I was wondering why are you using this fluorescent layer instead of direct laser excitation? If I got this correctly.
Yeah, so that fluorescent layer, we are now testing just with dyes, but the final idea is to have cells there. So if these cells, it's activated like a neuron, it becomes fluorescent, and then there will be a laser light generated that will go to the other cells and activate that cells. Yeah.
I see. So it's just like a test. At the moment, just the test system before you input the cells. Okay, thanks.
Yeah, yeah, right.
I have a quick question. It seems that you use a lot of laser illumination and so on. Does it somehow hamper cell viability during these experiments? Because this could be an issue with which we all microscopists, let's say, deal during these kind of experiments.
We think there is a possibility that there is a phototoxicity, but we never really tried yet, so we cannot tell by the data. But the laser dose we are using is in terms of irradiance. We thought that it's not that toxic level, so it's basically similar to confocal excitation. So for a certain range, it would be quite stable, but we need to test it.
Yeah, and how long are you going to illuminate? That's another point, basically, because if you expose cells for a long time and continuous illumination, that might be an issue. If you make pulses, might be okay.
So that's actually depend on the frequency of the output that light emitting part can generate. So we can use pulsed excitation. So we don't really use continuously pump the cells. So we can set it to like 10 hertz or 100 hertz depending on our needs. So for now, we're gonna try with low frequency excitation to avoid any cell damage.
Okay, thank you. Thank you.
I can foresee one rate limiting step, which is the optogenetics response after the laser. So even the laser light signaling might be fast, the optogenetic response still remains slow. So do you have any other ideas on how to speed things up? Because that would be your limiting factor.
So as you can see, we are actually integrating existing technology, and we are relying on the optogenetic actuator that people developed. So we found that so far C1V1 seems to be good, but if there is any good fast actuatable optogenetic actuator, we can adapt it. So for now, we are limited to current optogenetic actuators. We just started with intellectual curiosities, but one benefit would be our brain is limited by many biological constraints. One would be when you try to integrate more neurons into our brain, it's limited, and also the scale up is also limited by the transmission speed. So if we can actually develop this optical communication, we think we can unlimit the size of the brain. We may connect my brain to your brain. And this is just an idea.
Because of actually scholarship, there might be some limitations to using the optical accumulation anyway. I think it's the wrong way to go. But this
yeah, sorry, just a question that came up is, right now you're working with simplified version really with like one sender, one receiver. In the case, in the future, you want to extend that to more complex network, isn't there any possibility that there are crosstalks problem like with lights and stuff? And how will you be sure that you only like target specific receivers with your sender and stuff like that?
Yeah, of course, the final goal would be like single cell to single cell communication. Yeah, but due to like optics limitations, yeah, currently we are just using a bunch of cells in one and the other, because you cannot focus so much the light and so on. So there's some optical limitations, but yeah, the final step would be really single cell to single cell communication.
Thanks.
But if I may add one more thing, so one of the advantage of using the laser is it's really sharp peak compared to fluorescent signal. And then he can like excite or emit the laser in the different wavelengths. So in terms of the multiplexing, you can do the much larger, broader, the higher number of the multiplexing compared to the conventional fluorescence. So that's another advantage of using laser in addition to the really high power light emission. Yep.